So hello everyone, and welcome to this webinar about real-world handling of protein drugs. Happy Friday. Today, we have the pleasure to listen to Associate Professor Ulla Elofsson from RISE Research Institutes of Sweden and Associate Professor Mattias Paulsson from Uppsala University Hospital. My name is Daniel Peterson, and I'm the Director of Research and Business Development at the Department of Chemical and Pharmaceutical Safety at RISE. In order for you all to have the best experience of today's webinar, I kindly ask you to put your phones and mics on mute. Also, at the top right corner, you will have the speaker's view available. If you, perhaps it's already set, but you will get the best experience choosing this view, speaker's view. You are, of course, welcome to write your questions in the chat box. And after each speaker, we will take as many questions as possible. We are recording this webinar for your information, and this recording will be available at RISE webpage next week, as well as on our Pharma Office LinkedIn channel, Drug Development at RISE. And you're welcome to follow that channel for future webinars. All right, so that was a short introduction. So I will now welcome our first speaker, Ulla Elofsson. Could you please introduce yourself, Ulla? Welcome.
Yes. Hello, my name is Ulla Elofsson, and I will try to share my screen. Sorry. I'll do like this. There we go. So, good afternoon, everybody. My name is Ulla Elofsson, and I work as a senior scientist at RISE. And I'm also the coordinator of the IMI2 project, RealHOPE, which you will hear a little bit more about this afternoon. And the topic of my talk this afternoon is real-world handling of protein drugs, and it's based on a literature survey on that has been performed within the RealHOPE project. So I will first give a very brief perspective from the regulatory side, and then I will go through the handling steps that are involved in real-life handling, and which the people are, and the stressors that are connected to those steps. I will give a short summary, and then I will talk a little bit more about the RealHOPE project. So what about the regulatory perspective? There we go. So the regulatory requirements when it comes to in-use handling is not always so clear. So there is one guideline, for example, that is talking about compatibility. And here it is said that admixture or dilution of products prior to administration might need to be addressed. So it doesn't stipulate that it has to, but just that it might need to be addressed. And there is also guidance on stability summary, and there it says the summary should include, for example, conclusions with respect to storage conditions and shelf life, and if applicable, in-use storage conditions and shelf life. And in a recent commentary, the formulation work stream of the Bioform Development Group has presented a commentary that is based on a number of surveys around in-use stability and compatibility of biological drugs. And the results indicate that there was a diversity of practices between the different companies that answered the surveys. And the varied practices is due to partly that they use different molecular modalities and different routes and modes of administration. The different stability profiles that these products have, or the different therapeutic areas and clinical needs that they were designed for. So it might not be so hard to understand that the lack of guidance from the regulatory authorities might actually be due to these varieties that are found. So it might not be so easy to actually find a guidance that fit. It's not one size fits all in this case. So what needs to be considered when we are testing for in-use stability? So there is a lot of things going on. Once the protein drug has left the pharmaceutical company, it will be transported, and mainly by distributors. And it might also be along the way stored before it reaches the final destination of the hospital or pharmacy, where it again might be stored. And after that, it will also be prepared in some way, unless it's a ready-to-use syringe or cartridge or something like that. It needs to be compounded. That will be done by pharmacists or technicians or nurses. And after this, it will also be distributed throughout the hospital to the clinic or the ward where it's actually going to be administered. And the administration is, of course, done by pharmacists or nurses or physicians. So very many different people involved. And then there is also the patients and caregivers that pick up their medication at either at the pharmacy or at the hospital and transport it to their home site, where it will be stored quite often in their common ordinary cooler or fridges or perhaps freezers until it's finally used. And each of these steps are connected with different risks, as, for example, temperature, or out of range temperature, I should say. Light exposures, if it's perhaps left by the window, either in the hospital or at home, or outside somewhere on the way. Shaking and vibrations during transports of different kinds, shock if it's dropped, or it might also be pressure drops if that can happen during flights. And there are additional stresses as well, and that might be, for example, dilution. It's common that protein drugs are prepared by dilution, for example, into infusion bags, and this might lead to less optimal formulation properties due to that you actually dilute the excipients, protective excipients. You also have interaction with different interfaces and materials. And usually proteins are very prone to adsorb to different surfaces. And as long as they just sit there, it's most likely fine. nothing more will happen. But as soon as things are starting to stir, they might come loose again, and that might cause aggregation. And the different types of surfaces that might be involved are, for example, air-water interfaces. It might be oil-water interfaces, in particular silicone oil, which is used for lubrication and hydrophobization in different vials and different medical devices that are used. And these might also shed droplets into the solution, causing even more oil-water interfaces in this system. And there is also very many different types of materials in, for example, devices. And these might cause release of softeners or polymer monomers, which might affect the chemical and physical stability of the proteins. So if we first look at the distribution, distributors pick up the protein drugs products at the production site, and they transport them around the world, actually. They might also store them on the way. It might be stored by brokers, for example, and then it reaches the final destination of the hospital or the pharmacy. And during these transports and storages, the guidelines of the good distribution practice of medicinal products for human use, GDP, will apply. And that is a regulation framework that is quite thorough, and it keeps good track over, for example, the cold chain. And I think most of the people that are involved in this transportation is well aware about the importance of keeping the temperature range, the stability temperature range. Oops, sorry, wrong way. But what about, for example, light exposure or bumpy roads? Not so sure that that is so commonly known. And potential mishaps that can happen during transport is, for example, that a package is left out in the sun. And perhaps UV damage is not the highest concern here, because you have a secondary package around the primary packaging, which most likely shield from UV damaging. However, if you leave the package out on the tarmac, for example, say in Africa, you can imagine how hot it might be quite quickly. And so warming and thawing might be one matter of concern here. There might also be bad road conditions or turbulence during this transport, which causes shaking or increased interaction with, which can lead to increased interaction of air-water interfaces, for example. Dropped, particularly during loading and unloading. And this can cause impact that can lead to cavitation and local heating in the system. And the importance of the primary packaging was pointed out by Siska in a study by Siska and co-workers, which did shipping by public transport as well as lab simulations of dropping parcels. And they evaluated the number of particles that they found in the formulation. And they also found that the presence of polysorbate actually could protect this particular drug from aggregation under these circumstances. But they also pointed out that more research is needed to actually establish what type of impact will be detrimental to, or could be detrimental to and cause aggregation in protein drugs. The next step of distribution is the one within the hospital, and that I will leave to the next presenter, Mattias Paulsson, who will cover this part. It has in recent years become more common, I think, that you have care, home care, and also smaller clinics that might need prepared drugs that don't have the preparation unit themselves. So that means that ready-made or ready-prepared drugs need to be transported from the hospital to other sites. And this transport is not as regulated as the distribution from the pharmaceutical company. And it might also involve repackaging from the secondary package or even the primary package in some cases. And it might be very important that you pack your drug in a good way, which was pointed out in this study, where they investigated the shipping of syringes in boxes with ice packs. And what they found was that they used ice packs in order to keep the temperature for a long term. It was shipped by public transport. And the ice packs were good to keep the temperature around 0 degrees for about 30 hours, 25 to 30 hours. However, to begin with, they were kept way below in this temperature in the freezer. So when they first put them in the boxes and put the syringes in, the syringes actually, the content of the syringes actually became, the temperature became well below the freezing temperature within minutes. So it's important that you have control over the full time range, not only most of the time range. So this is, it's important how you do this. And it might also involve new types of transport devices, such as drones, for example, that has been suggested in recent years to be a good way of transporting medication to locations where it's difficult to reach by road, for example. Sorry. So healthcare and personnel, healthcare personnel. So in-use handling practices was recently investigated in a worldwide survey that was answered by 237 pharmacists from 37 different countries. And I would say perhaps only 92% reported that they stored the MABs in the right temperature range. And the rest was most likely due to that they didn't have control. So they didn't control the temperature, but they store them in fridges. But only 71% protected, ready-prepared products from light by secondary packaging. And very many used spikes or closed system transfer devices, needles, or a combination of these as medical device during the preparations. So there is a high level of variability in the daily practices of MABs in different pharmacies. And particularly on the questions on how monoclonal antibodies were transported after compounding within the institution, actually almost 10% stipulated that they use pneumatic tubing system. And this was studied by Carpenter, John Carpenter and co-workers quite recently. And they found that there were large increase of aggregate formation after the transporting from pneumatic tubing systems. And that the outcome depended on the type of bag material that was used and the type of diluent that was used. But they concluded that you should not tube your use pneumatic tubing for IV bags. However, there are different types of tubing systems used in the US and Europe. And there is also questions of whether the presence of headspace might actually affect the outcome of this. So patients and caregivers. Yeah, cold chain the cold chain is something that is important for all protein drugs. And in this study from Cleland et al., they found that there was cold chain breaches occur quite commonly, and particularly when they are stored, transported or stored by patients. So only 7% of the patients that participated in one study managed to store the medication within the correct range. And in another study, there was they found that the temperature was not logged. That the patients stored their medication. Only 20% of the participants store the medication within the recommended temperature conditions. So, however, the breach of the cold chain might not be due to the lack of understanding by the patients, but it might be that they don't have the means to control the temperature in their fridges. And they also might be due to that they have old fridges that might not be able to keep the temperature, which was shown in this particular study. So, in summary, more research into real-life events is needed, both within distribution in hospitals and home settings, and we need to know more about what is true and what is not in this case. And both professionals and patients, caregivers need more and better education in the handling of protein drugs so that you are aware of what you're doing. So now I'd like to tell you a little bit about the RealHOPE project. And this is an IMI2 project. It's within the partnership, private-public partnership of FPA and European Union. It's called RealHOPE, Real World Handling of Protein Drugs, Exploration, Evaluation and Education. And the project aims to build knowledge to ensure that protein drugs are safe and efficient during their whole life cycle. And the consortium of this project consists of 26 different partners from 6 European countries and the US. And we have 6 academic partners or research institutes, 4 hospitals with pharmacies, 1 pharmacy association, pharmacist association, 1 patient association, 1 instrument partner, and 1 app builder, and 2 distributors or logistics companies. And then we have 10 different FPA partners or partners in research. And we work to gain improved understanding of the real-life handling of protein drugs by generating, collecting, and analyzing data in real-world scenarios. And this we do, for example, by stakeholder interviews, investigational studies, and also by using electronic smart labels to measure the magnitude of different stresses. We're developing tools and methods for simulation of real-life events during the drug product handling that mimic the effects of handling in real life. And we aim to set up a miniaturized simulation methods for real-life handling. We also investigate how new technologies can be used for safer handling of protein drugs in hospital settings. And we also are working on developing educational materials tailored for different stakeholder needs. And I'd like to give a special acknowledgement to the people that has been involved in the compiling of the literature and the literature survey that I presented data from. And by that, I'd like to thank you all.
Thank you so much, Ulla, for a great presentation. So, dear listeners, do we have any questions? No questions?
I think, Georgia, did you raise your just a wave?
Ah.
Yeah, I was just congratulating you for the nice presentation.
I have a question, Daniel. You were very quick, but I was looking to find the different symbols. Now I've raised my hand, I think.
Excellent.
Thank you, Ulla, for a very nice presentation, I thought. And I've actually two questions, if that is okay. One was, I guess you're talking a lot about protein drugs because they are extra sensitive for stability. But is that true for all proteins, or would you say that there is a typical type of protein drugs where one needs to be extra careful, or how should one look at this?
You are on mute, Ulla.
Sorry, I was thrown out and came back in again, and that's probably muted myself. Yeah, so I think that you should treat, or you could think of all protein drugs as being quite sensitive. However, it has been shown that insulin, for example, that has been around for a long time, the insulin formulations of today are quite stable. So I think with time, good formulations can be developed, but it might take quite some time. So, and of course, different proteins, they're not all the same. So you can't treat them as all being all the same. You have to do formulation development for each and every one. But most of them are quite sensitive.
Mm-hmm. And another question, maybe a little bit similar type of question, but you also said that you were, as part of RealHOPE, like a very interesting consortium, you were going to set out to simulate this in-use stability. And then I was thinking, is there one type of stability experiment that would be extra useful, or do you need to actually simulate all the different type of uses that one has, like transport and infusion tubing and things like that?
So that is one part that we are looking into actually within RealHOPE. We are investigating what is actually happening in real life, and we will also be investigating the effect of different stressors and the sequence of the effect of sequence of different stressors. So we will hopefully know more by the end of the project about which type of simulations would be most useful to do and most important to do, I would say.
Thank you.
All right. Any more questions out there before we move on to Mattias? No? All right. Then I will now welcome our second speaker, Mattias Paulsson. Could you please introduce yourself?
Yes, hello everyone. Thanks for the invitation for this webinar. So I'm Mattias Paulsson. I'm a pharmacist. I have a PhD in formulations in pharmaceutics some 20 plus years ago, and I've been active in hospital pharmacy all the time. So I'm actually employed by the hospital, but I'm also affiliated to Uppsala University as a associate professor. So I will share some slides here based on one paper that we recently published around the protein drug supply chain within a hospital. So this is the actual article, and I was fortunate to have a large group of people around me from different countries, academia, hospital, and also the FPA partners here. So we got a lot of feedback from different perspectives. And those of you interested, you have the QR code to the right corner. So if you take your mobile camera and shoot on that, you will get the link directly to the paper. So the purpose of my talk is really to address this paper in more pictures. So I have went around with the camera. So that is what I'm going to talk about. And this is just a couple of slides of the hospital setting. So my university hospital is publicly funded It has around 900 beds, and Uppsala is a small town, so we get a lot of patients from other parts of Sweden, sometimes also abroad. In Sweden, we are unique in such a way that we have very few hospital beds per number of citizens. You can see in this rather old slide now, 5 years ago, that we have a mean in EU on 400 beds, and Sweden is in the end here. So this really puts a lot of stress on having active beds and a lot of logistics around. We try to do quite advanced home care treatment, and everything should be in place when it is needed. So this is kind of the context. So the method we choose to learn more about the actual real handling within the hospital was the Gemba walk method. And this goes back to Toyota and the lean manufacturing in Japan, where the management, the quality responsible people, would go and see and look at the actual flow of information or goods or knowledge and so on, and learn and try to improve all the time. So we went with this Gemba walk method to the different part, and everything starts with the distributor, the parts after pharma industry has delivered this to a wholesaler. And in our case in Uppsala, we have a collaboration with the company ApoX, which serves as a hospital pharmacy for a great number of hospitals around Stockholm and Uppsala and other parts as well. So we actually don't have a hospital pharmacy here in Uppsala. Instead, it sits some 1 hour drive, 70 kilometers away, just outside of Stockholm. So every day we get deliveries with a truck on road transport to my hospital here in Uppsala. And those of you who have been around, you know, Arlanda is right in the between there, the airport. So in order to learn about the logistics, me and the pharmacist colleague went to our friends at ApoX to look at the logistics there. How do they receive medications from different wholesalers, other companies, and how is this distributed further? And this is just one picture to show the boxes that is used with this on this conveyor belt. It's delivered with temperature control trucks like this to our consignee or the goods reception area. So this is actually in Uppsala, where it will end up on the other side of the wall here. So the porters come with this on carts, and you see the different boxes here. There's a system with cool boxes that are blue. You see the one on the right here, and the normal ones are the orange ones, and every one is marked with a barcode. So you have barcode scanning all the way what goods goes in these boxes, but also where they should go in the next step from this distributor, from the hospital pharmacy. And when it comes to Uppsala, well, you end up looking at different stresses. So this might be just a detail. It's not, I mean, these drugs have been designed to handle quite a lot of handling. But there are things you start to notice when you walk around. So that might be the first one you note. So then we go into the hospital and look at the transport all through the hospital. So from this scenario, we move on into the basement. So, you know, in Sweden, we have quite bad weather in the winter time. So you need to have underground transport to do this in a rational way. So from the goods reception area in the far left part of this map, you see there are quite a lot of distances. It's almost like a kilometer to reach the far end of psychiatry wards up here. And if you also take into account that the underground corridors are not straight, you end certainly up with more than a kilometer. And in order to have a rational way, the janitors would drive up and down and deliver to different units. So it it's a lot of driving underground with carts that would look something like this. And you see how this is handled. And this is, for example, one container with frozen medication. So it will be in such a styrofoam box. And then it's delivered, handed over to nurses or pharmacists in the ward. So you can't just leave it there. You need to have a person that will approve and sign the delivery. So then it ends up here outside the ward medication room. So to once again say, we don't have a centralized pharmacy. Instead, we have so many small pharmacies. Every ward has its own medication ward room where these medications are stored. So looking at this transport, we went on these electric cars. So we went, there are 2 seats on them. You can go and look how this is handled. And you see, once again, these kind of connections where there might be stresses on material in the carts. But what you really see that mistakes are done. So, I mean, we didn't spend that much time going around with this kind of janitors, but you see boxes like this dropped for different reasons. People are in a hurry and they pile up and something goes wrong and boxes are dropped. And I mean, if there is glass inside and the glass gets broken, something leaks out, this would typically be reported. If the janitor sends around a cake with the cream on top or something and a strawberry, and it will get destroyed, you can visually see that with your eyes. But handling of protein drugs in this way, it's not reported if you drop the box or if there is something else that breaks, if it's not obviously for this stuff that there could be a problem. So licensed formulations are likely developed from pharma industry to handle this kind of stress. But I guess it can't take too much of shock when it's exposed to continuous drops like this, if it happens. The next step in this Gemba walk would to look in the pharmacy and the clean room. So we have one compounding unit in the hospital. So it sits in the entrance 100 house. So it's this green area. So these are the clean rooms. They are right in the middle of the oncology ward. So we can easily supply the nurses in the treatment rooms all around here. They're so close to doing that. But on the same time, there is some distance to the pediatric oncology ward or to neurology and so on. So in some clinics, there is in-hospital transport of reconstituted drug. So inside the aseptic compounding, we have manual preparation, like Eva sits here and works in the cabinet. This is a B-class room with an A-class cabinet, safety bench. Or we also reconstitute with the Kiro oncology robot with the 2 reconstitution arms here, and you have built-in handling of you can use partially used vials. You can store them within this environment. And waste management and so on. You have barcode scanning, you have scales to check that the dose is right and so on. So there is a lot of nice tools for quality here. So looking at what kind of drugs we do within this unit, I made this table with the APIs here. You can see, for example, what APIs are available as pre-filled syringes. That is something that indicates that this is a stable, easy to use drug formulation. And you can see for some of these, there is readily available, but for many, there are no pre-filled syringes. So instead, the clean room needs to take a lyophilized powder, reconstitute it, or have a concentrate, like for these ones, and perhaps they dilute it, so they handle it. And sometimes many of these preparations are handled within the clean room. You see 100% of the preparations are handled through clean room, but in other cases, nothing is handled in the clean room. So then it's handled by nurses or in some cases, pharmacists out on the wards. So here you can start to look at what kind of wards are there and what kind of competence is there. But there is a difference now. We can start to see that the lowest risks are probably for pre-filled syringes that are developed to be able to being sent to a patient home and so on, kind of robust stuff. And then you have the ones that you need to reconstitute, you need to dilute and so on, that are likely more sensitive when you dilute also excipients and so on that are there to protect the drugs. So when stuff is made in the clean room, we have this kind of mailbox system. So nurses would come and open one of these drawers and access the preparation. And going back to this slide, you know, it sits right here. So they have easy access to all around here. But in the cases, the nurse is not having the patient in the same unit, it's transported once again with these electronic cars within the hospital. And sometimes we also have a neighboring hospital that does not have clean rooms. So we send to the neighboring hospitals. And in other parts of Sweden, you know, chemotherapy is always given rather close to the home. And we can't have clean rooms all over. So it's a lot of reconstituted drugs, infusion solutions that are handled and shipped to other sites. So this is not a big issue, but the truck or the airplane sending stuff to Gotland, for example, the big island outside here, could be something that needs to be addressed, how this is packed and so on, what kind of headspace is there in the bags and so on. So I would reckon there is a bigger risks for lyophilized drugs or drugs that are diluted post and then have a post-reconstitution transport, for example, trastuzumab. The in-hospital transport at our site would look like this. So when we actually go and discuss with the janitors, they say, oh, we get the infusion bags in a plastic bag like this, and we put it on our small, what do you call it, scooter or something. And I can't say that I think this is sufficient. We need to work on this to have an improved container, something that is polystyrene or something that takes the vibrations away. I don't think this is a good system. And this is what it looks like in my place with some knowledge around this. In the ward, we have a situation looking like this. So nurses often prepare outside and vial sharing takes place all the time in the clean room, perhaps not as common in the wards, but it may be there because many of these drugs are so costly for us. So we really like to share vials, and then you need to think of the aseptics, the storage time and so on. In Sweden, it's 12 hours you can use an open container like this injection vial when it is reconstituted in the ward. But in a clean room, you could for sure have up to a week of shelf life using vials if there is data. Also in this environment, in the ward, you have a computer with a order, a prescription from the doctor. But what has really come into that order? What is the necessary information? Does that prescription really say what is the right choice of infusion solution? Glucose dextrose, sodium chloride, sterile water, and so on. How has that gotten in? So that's also a risk around that to have a approved template in the prescription software. A lot of decisions around this also has to do with the hazardous material. So do we work with the CSTD like Ulla mentioned before? That is based on the hazardous risk assessment. And I just wanted to share this document with you. It came last week, this is fresh from the press. So those of you interested in occupational health issues, please have a look at this document. So CSTD is just some reference, the American NIOSH and some examples where you have a closed system drug transfer device in order to prevent occupational health issues with the workers. But that can also introduce shear forces, absorption to surfaces, and so on that we previously discussed. So my last part of this Gemba walk would be the one very close to the patient and the administration. So this could look in different ways depending on the setting, of course, but a typical oncology daycare polyclinical setting, you would only administer one infusion at a time. But in the ICU, perhaps in pediatric oncology or other settings, these patients have so many medications, could be like 10, 15, 20 medications, and some of them are needed to be given simultaneously. And then you end up with something looking like this. You see this where you have multiple connections. So you spike the infusion bag up here and you can connect different pumps to that. You have the drip chamber. You can see here it mixes before it goes into the patient. You need data about this. Can these multiple drugs be mixed in a safe way? And there's very little documentation out there on that. Perhaps there is a dead volume somewhere that this bag is not completely emptied. So you end up with having some drug dose up there. So it's a lot of practical things that we perhaps are not aware of unless we go with a Gemba walk and see what actually happens. So based on the findings we did, we made this FMEA and identified the risks that I have talked about now with you. And we took the risks, we looked with the double diamond method to see how can we mitigate it, what kind of protocols could we use, and how can this be improved. And we did simulations to see that a lot of the problems could be mitigated. So, I mean, distributor, we have a GDP service. So I think it's very controlled the way the substances comes to us, but still in the post distribution part, there could be issues, dropping of boxes and so on, that could happen. And around the patient administration, that is still rather hard to control completely. So those were the slides I had for today, and I end there because I would very much like to hear some opinions and some questions from you.
Thank you so much, Mattias, for a lovely presentation. All right, any questions, dear listeners? Yes, Co.
Yes, Mattias, thank you very much for a very nice walk through the hospital.
Yeah.
So I was wondering how representative do you think the hospital in Uppsala actually is for other hospitals?
Yes, that's a good question. I mean, I think it's rather unique, the Swedish way that we have the hospital pharmacy as in a different site, 70 kilometers away. That's, I think, unique for Europe. We don't see that in many places. But the janitor work and this the handling the chemotherapy robot, a lot of things are similar, and we actually have a collaboration with Barcelona. So we are comparing the two different sites with the hospital clinic in Barcelona to see what are the differences, because we want to make this more generalizable so that more can see how and recognize this in their own setting and start. I want to inspire people, pharmacists around Europe, to leave the pharmacy and go out on the wards. There are many people that never leave the pharmacy to understand where the big problems really are. They are perhaps 8.4 or 8.3 degrees centigrade is a big difference, they think, in the pharmacy, and they have no idea what goes wrong up on the ward. So that, I think, is important.
that also brings me a little bit to my other question. And when looking at all the different things that can happen, people drop boxes and you have a lot of pumps that you can enter, or dead volumes in infusion bags and things. I mean, I can see that these are really important for the stability, but what will you do with the information that you get from that? But what will you do with the outcome of this?
Yes. We gather a lot of information, and of course you can say you shouldn't do that and you shouldn't do that. You can write a lot of documentation, but there's so plentiful of information out there. So we need really to try to get some information in the templates for the doctor, so it follows the prescription, how it should be handled, what filters to be used, how to handle the medication. And then I think, I mean, we have temp tails for temperature. Perhaps we would need something to also check shock, and if a box has been dropped, there should be some indicator to see if you have some drug product that is really sensitive, that you can see that this box must have been dropped. It's not just the temperature, it's also if it has been tilted or dropped, that you can see things like that. To build in, we write it in the paper also, to build in using technology instead of writing instructions. I don't think that's successful. We should more rely on technology, I think, for this.
Thank you.
All right. Any more questions out there to Mattias? No? All right. So then, yeah, it's 3 minutes left of this webinar. But if you don't have any more questions to Mattias or Ulla, for that sense, it's time to finish this one off. And so thank you all for listening. And we wish you all a very nice weekend. Thank you.